All right, testing. All right, so let us begin with the nervous system. All right, so today we're going to be talking about nervous tissue, the basic structure and function of nervous tissue. So, so nervous tissue is made up of two cell types, neurons and neuroglia. Neurons are the cells that actually conduct nervous impulses, and they can carry those impulses to distant points, or another phrasing is points remote from the cell body. So if you look at the um, neuron or neurons that control, say, the pronator teres muscle in the forearm, okay, the cell body for that neuron, or those neurons, because there will be more than one, is actually located in the spinal cord. And so the axon, which carries the signal away from the cell body, is going to have to travel out of the spinal cord along the path of some spinal nerves down through the brachial complex and into the brachial region, and then down into the antebrachial region, and then finally to the pronator teres muscle. All right, so if you were mapping that on your body as we went through it, the distance from the spinal cord in the cervical and thoracic region down to the pronator teres muscle, depending on your height and body size, could be about 1.5 to 2 feet, you know? So just in that one example, this one cell is quite long, and it's carrying this nerve impulse or action potential all the way down that length, all the way down the 1.5 to 2 feet it takes to reach the pronator teres muscle. All right, so neurons typically react to a stimulus, and this can be a physical or a chemical change in their environment. We'll talk about receptors in the senses unit a bit more. And then neuroglia are cells that support the neurons, right? And so the example I always use is, most people probably think that most people in the Air Force are flyers. And the fact of the matter is, only some small percentage, maybe 25%, maybe 10% of the Air Force actually flies. The rest of the Air Force is performing functions that support the flyers. So things like, I mean, yeah, there are people in human resources. There are doctors who are in the Air Force. There are people who maintain and repair aircraft. There are people who fuel aircraft. There are military police who protect the base. So there are a lot of people in the Air Force who are not flyers, but who are working to support the mission of the flyers. All right. And so that's kind of what we've got going on here in the nervous system. We have neurons who are doing the actual work of conducting these impulses, what you think of when you think of the nervous system. And then you also have the neuroglia who are supporting the neurons, and the majority of cells in the nervous system are actually neuroglia, the support cells. They're also known as glial cells. We'll talk about them more in just a little bit. And then nerves are bundles of axons, and we've got a pretty good picture. It's similar to the organization of muscle. We'll talk about that. So nerves are actually not cells. When we talk about nerve cells, this is kind of a misnomer and it's a bad name, but everybody uses it from time to time. Nerve cells, when we say that phrase, we're referring to neurons. It's kind of a bad name. We should probably not use the phrase nerve cells, but everyone does. So a nerve is a different structure. All right, so here we're looking at a neuron. This is a multipolar neuron. We'll talk about neuron classification in just a little bit. This is kind of the stereotypical cartoon image of a neuron. When people draw neurons, they almost always look like this. They almost always are multipolar neurons, and We'll talk about the other types of neurons in just a little bit, but here we have a couple important structures. So the main part of the cell here, the largest part, well, largest in diameter, is the cell body, also known as the neurosoma or soma, S-O-M-A. And coming off of the soma are the dendrites. And the dendrites receive signals from either sensor cells or they receive information from other neurons at the dendrites. So the neuron receives information from wherever, whether that's another neuron or some sort of sensory cell, receives information at the dendrites. Okay. Now that information results in potentials that are going to initiate an action potential in this region known as the axon hillock. This region is the axon hillock, and this is where action potentials actually begin in the neuron. Here we have this long cylindrical process, this long thin process coming off, and that's the axon. The axon carries the action potential away from the neurosoma. In other words, it carries that action potential on the path to whatever the target cell is. Now, that target cell could be muscle, whether that's skeletal, smooth, or cardiac muscle, or it could be a gland. All right. The target's always going to be either some form of muscle or a gland, right? The targets are also known as effectors, and we'll talk about that when we get to reflex arcs. All right, so here we have, yes, the axon carrying that information away, and then from the muscle unit, you remember that at the end, it's not pictured here, but at the end, we have this terminal enlargement of the axon, known as the terminal button, end button, synaptic knob, end knob. Physiologists like to use the term bouton. So anyway, it's got a lot of names, but that swelling that we saw that formed a synapse with the motor end plate, we're going to have that same swelling wherever we form a synapse with the target, whether that's skeletal, cardiac, or smooth muscle, or glands. All right, then we see a couple glial cells over here. So this is an astrocyte. That's one of the types of glial cells, and it forms part of the blood-brain barrier, and its job is largely to regulate the chemical environment in the central nervous system. It also provides bulk, it fills in space in the brain. Most of the cells in the brain are actually astrocytes, and in the condition multiple sclerosis, where we form scar tissue in the brain, that'll be astrocytes migrating into dead or hollowed out empty regions of the brain and then filling them in, forming these scars that we can see on a MRI or CAT scan. Then over here we have the microglial cells. Microglia are going to function to remove pathogens from the central nervous system. All right, so that was just one example. Good, we've talked about a lot of this already, but the neurons are, yes, also known as nerve cells, although that's really not a very good name because I think that's confusing for students. All right, so you will find the axons of neurons in a nerve, but the nerve is not a cell. The nerve is a larger structure made up of multiple axons. But anyway, these neurons, they detect stimuli. It's a very common phrase, by the way. So, I mean, you can use it. It's just, it's sort of inaccurate. So they detect stimuli, they respond quickly, and they transmit coded information rapidly to other cells. At the beginning of ANP2, well, actually, different professors handle this differently, but most professors begin ANP2 with endocrine. I know at JCTC there are a number who don't, but and that's up to each individual. Some feel that you need to cover blood and the circulatory system before you cover the endocrine system, but I always start with the endocrine system. And if you compare the nervous system to the endocrine system is the system of glands responsible for producing hormones in the body. If you compare those two systems, both are used to transmit messages throughout the body, but nervous tissue transmits messages much more quickly, and then the response is done as soon as we're done sending action potentials. So the stimulus, as soon as it ends, the response ends. If you compare that to the endocrine system, it takes a lot longer to produce hormones and release them. It takes a lot longer for hormones to circulate throughout the blood. And then you can stop secreting hormones, but they're still in the blood and the tissues. So turning off that response is going to take longer in the endocrine system. All right, so we mentioned the soma or neurosoma. This is the cell body where you find the nucleus and the organelles. And we actually produce neurotransmitters in the neurosoma, and then they have to be carried down the length of the axon to the bouton or synaptic knob or whatever you want to call that. It has like 5 names. Um, Dendrites we mentioned. These are the branching processes that come off of the neurosoma, and they receive signals from other neurons or receptor cells. And then the neuroglia, we mentioned they protect and assist the neurons. All right. So the word dendro, or the prefix dendro, is Greek for tree, and so these are like the tree-like processes that come off of the neurosoma. All right. And this is the primary site for receiving signals from other neurons and other types of sensory cells. Some neurons are going to have only one dendrite, but some may have thousands. And the more dendrites a neuron has, the more information it can receive and incorporate into its decision making. So down here we see two multipolar neurons, and they are in contact with one another. Here we have the first multipolar neuron and its cell body or neurosoma. And when it initiates an action potential, it'll do so here at the axon hillock. Then it travels down the axon. Okay. All right, and then we have the terminal boutons, synaptic knobs, whatever you want to call them. They're in contact with the dendrites on the target neuron or on the second neuron. Okay, and so the second neuron is receiving a stimulus. It's receiving a signal from the first neuron at its dendrites. Okay. And then if the stimulus is big enough to initiate an action potential in the second neuron, that'll of course be initiated at the axon hillock and then travel down the length of its axon to whatever its target cell is. I just want to talk about decision making real quick. This is going to be a very, very, very basic example, but all right. So let's say you have a neuron. Let's say it's as simple as that. You've got one neuron that controls all of the muscles uh, that move the body in such a way that you step on the gas pedal, right? All right. Controls all the muscles of the leg that allow you to step on the gas. All right. So when does this neuron fire? Well, it's got dendrites here. All right. When does this neuron fire? Well, there's another neuron over here. And it's hooked up to the eyes somehow. Okay. And it sees that the traffic light has turned green. Okay. So the eyes detect that the traffic light has turned green. This neuron in the brain interprets that information, and then it's going to initiate an action potential that travels down the axon and is received by the dendrites of the second neuron. It's received by the dendrites of the gas pedal muscle neuron, right? And so if this neuron is sufficiently stimulated, an action potential will travel down. Actually, let's do this in green. Will travel down the length of the axon and stimulate the muscle fibers that are responsible for stepping on the gas pedal. Okay? And so that's really simple decision making. Basically, we saw the light was green, so we stimulate the motor fiber that stimulates these muscle fibers that step on the gas pedal. Okay, now let's make it a little more complicated. We've got another neuron here. All right, and it's going to have its own terminal arborization, its own end bulbs, synaptic bulbs, boutons, whatever, interacting with the dendrites of the motor neuron. I'm going to write motor neuron there. All right. It has its own dendrites. All right. And I'm going to say those are hooked up to the eyes also. And they're looking out for pedestrians. And bicyclists. And so basically, if it sees pedestrians, it's going to tell the, sorry, get my mouse back here. It's going to tell the motor neuron, don't go. Okay. And so its action potential is going to travel down the length of its axon, and it's going to say, don't go. It has an inhibitory effect. The green neuron over here says go. Okay. And so if the neuron on the left sees the light as green and there are no pedestrians, then it's going to stimulate the motor neuron to go. If, on the other hand, the eyes see the light has turned green, but there are pedestrians, the green neuron will stimulate the motor neuron, and the pedestrian neuron over here will inhibit the motor neuron. Okay? And we can actually do that. So the green motor neuron would release stimulatory neurotransmitters, and the pedestrian motor neuron would release inhibitory motor neurons. I'm sorry, inhibitory neurotransmitters. All right. And so by balancing the strength of the stimulus from all of the neurons that interact with the motor neuron, we either are able to reach threshold, depolarize, and initiate an action potential, or we are not able to do that. We can block that action potential with the inhibitory neurotransmitters from the pedestrian's neuron. All right, I hope that was clear. It's just an example of how we make decisions. Oversimplified. Of course, there's a whole lot more going on than just is the light green and are there pedestrians. All right. So, structure of a neuron. We mentioned the axon hillock. That's this swelling of the axon here. It begins and gives rise to the axon itself. The axon is this long cylindrical process that can travel very far away from the soma or neurosoma or cell body. As we said in the example of the neurons that innervate the pronator teres muscle in the forearm, that can be a distance of 1.5 feet, 2 feet. If you're one of these U of L basketball players, probably 2.5 feet. right? Big wingspan. The muscles, I'm sorry, the neurons that innervate the muscles of the leg have to travel even further, right? So from the spinal cord at the level of about L3, L4, um, they'll exit the spinal cord and then they'll have to travel out and down. I'm looking at my own leg and my torso here, and I'm estimating that's a distance of maybe 3 feet that the neurons axon would have to travel to reach some of the muscles in the lower limb. All right, so yeah, this axon can be pretty long, can travel to places remote or distant from the neurosoma. All right. So one of the key features of a neuron is actually that the signals are able to be conducted rapidly, right? And so if you look at that distance of 3 feet, you'd never be able to tell, right, that the signal had traveled that long, that it had traveled 3 feet from the spinal cord. Um, it seems to us to be either instantaneous or almost instantaneous when we tell the muscles of the leg to contract. It happens pretty much immediately, and that's because the action potential is able to propagate or travel down the axon very quickly. And that happens actually because we have this myelin sheath. And so you've seen this drawn in a couple of cartoons so far, although we haven't pointed to it. You see these little regions of insulation surrounding the axon. Okay, in the peripheral nervous system, that is in the nerves and neurons that will travel out of the spinal cord to the rest of the body, you're going to find these Schwann cells. They create a sheath around the axon called the myelin sheath, and it's about, I want to say it's about 80% lipid, about 20% protein. And what's really interesting about it is that it leaves these gaps. Okay, and these gaps are called nodes of Ranvier, French name Ranvier. And so basically, the gaps or the nodes of Ranvier are actually what allow the signal to be propagated more quickly. By depolarizing this region near the axon hillock, we initiate the action potential. And then instead of having to open and close sodium and potassium gates all the way down the axon, we actually can flip the polarity in this region, depolarize it, and that'll open up voltage gated sodium channels in this region. We flip the polarity, depolarize it, and that is enough to open up the voltage gated sodium channels in the next node of Ranvier and the next, and so on down. And so the signal is actually able to sort of skip these regions, which greatly speeds up the rate at which we're able to conduct this message along the axon. All right. So good. Oh, and so just like the muscle cells, we had the sarcoplasm, which was the cytoplasm of the cell, and we had the plasma membrane called the sarcolemma. We're going to have axoplasm, is the special name for the specialized cytoplasm of the axon. And then the axolemma is the name for the specialized plasma membrane of the axon. All right, so at the distal end, you have what's called a terminal arborization. This could be confusing because arbor is Latin for tree. Remember, dendro was Greek for tree. Arbor is Latin for tree. We have Arbor Day. It's the day where we go out and plant trees. Some people do. And so you get these extensive fine branches that come off of the end of the axon. So this also looks like a branching tree pattern. One way maybe that you can remember it is, all right, dendro up here is Greek. And that's at the beginning of the axon, right? That's where we receive our signal. We initiate a new signal, travels down the axon, and then reaches the terminal arborization at the distal end of the neuron. One way to maybe remember that, if you can, is that dendro is Greek, arbor is Latin, and the Greeks came before the Romans who spoke Latin, right? So the Greeks rose up before the Romans did. All right, Greek before Latin. Anyway, not super important to remember that. Should know for sure dendrites. Terminal arborization is a term that may or may not come up again. Good, so they all end at a synaptic knob, also called an end knob, terminal button, bouton, B-O-U-T-O-N. Lots of different names for this swelling, this structure at the end of the terminal arborization. And this terminal arborization ends, yes, in the bouton, the synaptic knob. I think I'm going to try and stick with that term, synaptic knob. And it's going to form a synapse with the target cell. Okay, the target cell for a neuron is always going to be either another neuron or a muscle cell or a gland. Okay. And in the synaptic knob, we're going to find all these tiny little vesicles, those little membrane bound sacs full of neurotransmitter. And you remember that when the action potential, that wave of depolarization, travels down the axon, it reaches the synaptic knob, and then in this region opens up voltage-gated calcium channels. The calcium floods in, binds to proteins on the vesicle, and then the vesicle is able to fuse with the membrane here and dump its contents out into the synapse. Now, actually I should say synaptic cleft. This image is mislabeled here. It says this region is the synapse. This is actually the synaptic cleft. The space between the synaptic knob and the target cell is the synaptic cleft. The synapse is actually this overall structure, this junction between the neuron and its target. All right, so the neurotransmitter, just like in the muscle unit, has to diffuse across the synaptic cleft and then bind to receptors, all right, on the target cell, whatever that target cell might be. All right, so here we have structural differences between neuron types. And I mentioned multipolar neurons a couple of times. When people draw a neuron, it's almost always a multipolar neuron. I don't know why that is. Maybe they're the most interesting looking or the easiest to understand for us. It's just one of those weird things. People always choose to draw multipolar neurons, even in situations where you probably wouldn't be looking at a multipolar neuron. Textbooks do it too, all the time. Textbook images will draw a multipolar neuron where really you'd probably be looking at a bipolar or a unipolar neuron. All right, so why is the multipolar neuron multipolar? Well, it has one axon coming off, but it's got several dendrites coming off, and so it's got multiple poles. So we say it's multipolar. Multipolar neurons, it says right here, are going to be motor neurons most of the time. And so the neurons that control the muscles in the body, these are going to be multipolar neurons that have a cell body in the spinal cord and an axon that travels down out of the spinal cord and then to the muscles of the body. We also have bipolar neurons, which have two poles. You can see two processes coming off. Okay, they have one axon technically and one dendrite. All right, one of these processes is technically a single axon. The other process is technically a single dendrite. All right, and so these are going to be interneurons or, well, yeah, interneurons. You also have the olfactory cells in the nasal cavity, certain neurons in the retina, and sensory neurons in the inner ear. In the spinal cord, we'll see interneurons connect neurons in the spinal cord. Basically, when one neuron needs to transmit a signal to another in the spinal cord, we'll be looking at interneurons. They're an important part of the reflex arc, and we'll come back to those. Next up, we have unipolar neurons. These tend to be sensory neurons. And as you can see, there's one process that comes off, and then it sort of branches in two directions. One direction is going to be the dendrites, the other is going to be the axon carrying the signal away. But technically, there's one process coming off, so it's a unipolar neuron. And these are going to be the sensory neurons that carry information from the sensors around the body to the spinal cord. All right, so basically, if you feel cold, right, your body feels cold, you're feeling that in your skin. The temperature receptors or thermoreceptors are located in the skin, and they're going to stimulate the dendrites of a unipolar neuron, which carries that information all the way back to the spinal cord. And then in the spinal cord, it will stimulate another neuron that carries that information up to the brain where it can be processed. Pain receptors also. Most of the time when you feel pain, you're feeling that in the skin or maybe in the fascia. And so those pain receptors, they detect that stimulus, they carry it, yes, to the spinal cord, and then they stimulate another neuron, possibly an interneuron, which will notify the brain and form typically a reflex arc with a multipolar neuron. But we'll talk about reflex arcs in a little bit. It's just better with the pictures in front of us. And then you have anaxonic neurons, such as the pyramidal cells. My headset turned off. Sorry. Let me get the charging cable just in case it's dying. Let me hook that up. All right, yes, sorry, that seems to have been the case because plugging it in, well, it died while I was trying to find the charging cable. It's working great now. All right, so yeah, the anaxonic. An means without or not. So there's no axon for these anaxonic neurons. That includes the pyramidal cells, which are located largely in the cerebral cortex. All right. And so they actually communicate through their dendrites, but they don't produce action potentials. Some anaxonic neurons are found in the brain, especially in the cerebral cortex, the retina, and the adrenal medulla. All right. So here's how axonal transport works. Things are produced in the neurosoma. Neurotransmitters, proteins, whatever, it's all made in the neurosoma. And it has to be carried down the axon to the synaptic knobs, right? Especially vesicles. We talk about vesicles. That has to be produced in the soma and then carried down. All right, the neurotransmitter. I'm not sure if I said synaptic vesicle or not, but the neurotransmitter has to be produced in the neurosoma, carried down in vesicles, and then the vesicles wait here until the action potential arrives and they can dump out the neurotransmitter. They will also take it back up. That's called reuptake. And we talked about in the muscle unit an enzyme called acetylcholine esterase. Acetylcholine esterase sits in the synaptic cleft and it breaks down acetylcholine to help end the stimulus, to help end the stimulation of the muscle fiber. All right, so it breaks down or decomposes acetylcholine, and the byproducts, the waste products, are taken back up by the synaptic knob and they have to be transported back to the neurosoma to get recycled and made into new acetylcholine. All right, so there's two types of transport. We are traveling along protein fibers located in the axon, and there's anterograde transport. Anti means before or in front, and so anterograde transport is transport forward down the axon. Retrograde transport, retro is like back in the past, right? It's a throwback, retro. Retrograde transport is where we are transporting things back up the axon to the cell body. And this is performed by two proteins known as kinesin and dynein. Kinesin is responsible for anterograde transport. Dynein is the way back, the protein responsible for carrying things back to the neurosoma. And the mnemonic I used when I was a student was KD. So if you play Halo, and I know fewer and fewer students every semester do play Halo, but uh, others play Call of Duty or other, FPS games. And so in those games, you have a statistic called the KD ratio. That's kills versus deaths for the match. And so as a student, I remembered that KD, kills deaths. And then I applied that here. Kinesin, dynein. Kinesin is responsible for anterograde transport, transporting things down the axon. Dynein is responsible for carrying things back up the axon. If that doesn't help, there are probably other mnemonics out there. That's just the one that worked for me when I was a student. Good. So, yes, note the organization. I added this last semester. The organization is remarkably similar to skeletal muscle, right? So if we're looking at this, we're looking at the anatomy of a nerve. This is an actual nerve here, all right? Just like we had the large anatomical muscle, that's going to be similar to a nerve here, although the nerve is much, much smaller. We start down at the most basic. Let's do that. You've got a single axon here. Now the axon is wrapped in Schwann cells forming that myelin sheath that we talked about, that insulating layer called the myelin sheath. Now, each individual axon, by the way, the axon is also known as a nerve fiber, right? So the individual muscle cell was a muscle fiber. The individual axon, although it's just part of a cell, it's not the entire cell, it is the nerve fiber. All right. And each nerve fiber or each axon is wrapped up in a connective tissue called the endoneurium, just like we had the endomysium in muscle. Now, multiple nerve fibers or axons are wrapped up into a bundle called a fascicle. Just like in muscle, multiple muscle fibers were wrapped up in a bundle called a fascicle as well. And in the muscle, it was wrapped by a connective tissue layer called the perimysium. Here in the nerve, it's called the perineurium. All right. And so if muscle has endomysium, perimysium, and then epimysium surrounding the anatomical muscle, in the nerve, we're going to find endoneurium surrounding the individual nerve fibers. Fascicles are wrapped in perineurium, and then multiple fascicles will be bundled together and surrounded in epineurium. All right. And so the nomenclature here and the organization is remarkably similar, yes, to muscle. This is a cross section of a nerve, if that wasn't clear. All right. So the nervous system is divided into two large parts to begin with. We have the central nervous system, which includes the brain and the spinal cord. All right, that's it, brain and spinal cord. That's the central nervous system. The peripheral nervous system includes the nerves and ganglia outside of the spinal cord. And so all of these nerves that you see running out through the body to all of the target tissues, in the femoral region here, in the sural and crural region, down through the pedal region, right? Just for example, all of these nerves running through the rest of the body, This is the peripheral nervous system, and that includes these bulging swellings next to the spinal cord. Those are known as the ganglia plural or ganglion singular. And textbooks always call it a knot-like swelling. I think that's a weird way to describe it. It's just this enlargement next to the spinal cord. And that's where the cell bodies of a lot of neurons are going to be located, especially in the autonomic nervous system, which is a further division of the nervous system we'll talk about in a little bit. An important note, the peripheral nervous system, or PNS, includes both the cranial and spinal nerves. And so we will talk about cranial nerves. These are the nerves that originate in the brain. Well, one of them comes from the spinal cord, but mostly through the brain and central nervous system here. And they act within, most of them act within the head. There's one of them, cranial nerve 10, the vagus nerve, which actually travels down through a lot of the body. But we'll talk about that when we get to it. Cranial and spinal nerves. The spinal nerves, there are a lot of them. These are spinal nerves traveling throughout the body. These are all spinal nerves that we see here in yellow. The cranial nerves each have individual names, but we'll cover that. You really need pictures for all this. All right, so the sense organs and simple sensory nerve endings receive information about changes in the body and the external environment and transmits coded messages to the central nervous system. The central nervous system, or CNS, processes this information and relates it to past experiences, and then it determines what response, if any, is going to be appropriate to the circumstances. It then, if it determines a response is necessary, issues commands, primarily to muscle and gland cells, to carry out such a response. And so this is an oversimplified view of a reflex arc. So here you have a head and it sees water. Maybe the body is thirsty and it knows from past experience that drinking water alleviates thirst. All right, so that sensory information is carried through sensory neurons to the central nervous system. Now, of course, because this information is carried over cranial nerves, that's part of the peripheral nervous system. That sight is carried over cranial nerves, the optic nerve, back to the brain, okay, where the information is integrated. Integration is where we take all of the sensory input, compare it to past experiences, and decide what response, if any, is necessary. If a response is necessary, then you will see motor output here. This is where we are stimulating motor neurons to grasp the glass and lift it to the mouth so that we can drink. The sensory input is vision. We see the glass. We integrate that information. So in this decision making, we'll integrate not just our visual localization of the glass, not just that we see where it is, but also the current position of the body, because we need to know that before we can tell the body how to move in order to reach for the glass. We then send that information out to the effectors, in this case, skeletal muscle that will grasp the glass and lift it to the mouth. All right, so the sensory or afferent division of the nervous system carries signals from receptors, again, the sense organs and simple sensory nerve endings, to the central nervous system. We have sense organs such as the eyes, which are highly complex structures. We also have some simple sensory nerve endings, such as like pain and itch receptors embedded in the epidermis of the skin. All right, so the somatic sensory division carries signals from the receptors in the skin, muscles, bones, and joints. Basically anything that you're consciously aware of, pain coming from the skin, muscles, bones, and joints, to the brain or central nervous system here. Doesn't necessarily have to be the brain, by the way. The spinal cord is actually capable of making some decisions on its own without the brain. And then the visceral sensory division Visceral, remember, refers to the organs, carries signals mainly from the viscera of the thoracic and abdominal cavities to the brain or spinal cord, and that information will be acted on by the central nervous system. We are typically not aware of visceral sensory information. We're aware of somatic sensory information. We're aware that our skin is cold, our muscles are sore, our bones are broken, our joints are overstretched or inflamed. These things we're aware of, right? But think about it. All right, this morning you ate a breakfast burrito, and tonight that has traveled all the way through your digestive system and it's wound up at the other end. How'd it get there? Do you remember feeling it move at all once it left the esophagus and entered the stomach? Do you remember feeling it move at all after that? No. The stretch information is detected by receptors in the wall of the stomach and the small intestine and the large intestine. And unless it's fairly painful, unless it's extreme, you're typically not aware of that information. It's carried through the visceral sensory division, and you are not aware of exactly where in the digestive tract your breakfast burrito is right now. Not until it's ready to come out. Then you'll be aware of that usually. All right, so the motor or efferent division of the nervous system carries signals from the central nervous system and the muscle cells, also known as effectors, gland and muscle cells, also known as effectors, that carry out the body's responses. Another way of phrasing this, the effectors are the cells that carry out responses on behalf of the central nervous system. These effectors are going to be either glands or muscle cells. Doesn't have to be skeletal muscle, by the way. In the visceral division, we can control smooth muscle as we do in the GI tract or digestive tract. Now here we have the somatic motor division, and again the somatic nervous system is the portion of the nervous system that we are consciously aware of or can consciously control through the somatic motor division. This is going to be skeletal muscle, and that's pretty much it. Yep. You cannot consciously control glands, you cannot consciously control cardiac muscle, and you cannot consciously control smooth muscle. Only skeletal muscle can be controlled through the somatic motor division. All right. So we control, yes, the muscular contractions, the skeletal muscle contractions. That's under voluntary control. We choose to move certain ways. Now, skeletal muscle will never contract unless stimulated. That doesn't necessarily mean that just because we can consciously control skeletal muscle that we are always choosing to make movements or contractions with the skeletal muscle. So you have somatic reflexes, and these are things that you cannot control. This is involuntary control. And an obvious example of this is you've all been to the doctor at some point, I'm sure, and he or she has hit your patellar tendon with that tiny triangular hammer, and then your knee jerked out, right? That knee jerk reflex. In order to produce that movement, that knee jerk, you had to stimulate skeletal muscle, but you didn't make the conscious decision to do that. It's part of a reflex arc that involves the spinal cord, not the brain. Oops. And so anyway, not always. Just because the somatic motor division allows you to consciously control muscle doesn't mean that all skeletal muscle movements will be voluntary. You have somatic reflexes, which you cannot control. And then there are also neurological disorders which cause spasms and twitching that you are not consciously initiating in the skeletal muscle. All right, then we have the visceral motor division, also known as the autonomic nervous system. Um, and so basically, it's going to carry signals to the glands, cardiac muscle, and smooth muscle to perform visceral reflexes. These are, of course, unconscious. You are not aware of them. So I mentioned that breakfast burrito you had for breakfast, or whatever it was, protein bar, and you swallow it. Somewhere in the esophagus, you stop sensing it, and then later tonight or tomorrow, suddenly it arrives at the other end of the digestive tract. It was carried there by rhythmic contractions of the smooth muscle in the walls of the GI tract. This process of performing the rhythmic contractions is known as peristalsis, and it propels food through the digestive tract. All right, travels all the way through thanks to this peristalsis, this rhythmic contraction. And you weren't aware of any of it. You didn't consciously choose to do that. All right, so the autonomic nervous system is further divided into the sympathetic and parasympathetic divisions. Now, the sympathetic division, it says, tends to arouse the body for action. Arouse in this case means to awaken or stimulate. The parasympathetic division tends to have a calming effect. Now, the sympathetic division is also known as your fight or flight division. So when you are walking, you turn the corner and you see a saber-toothed tiger or a mugger or something like that that frightens you. A zombie, right? Um, any of these things, or just an unexpected person. Uh, they activate the sympathetic division, and that's going to prepare the body to either fight or flee, run away. Yeah. In either case, we're going to need similar responses. We're going to need to increase blood flow to the muscle. We're going to need to stop digestion. We're going to need to dilate the irises so that we can take in more light and see more detail. You know, um, so we're going to need to do all those things. The parasympathetic division is also known as the rest and digest division. And so basically, when we are not in danger, when we do not perceive a threat, when we are not stressed, the parasympathetic division will take over and it will tell basically the heart rate to go ahead and slow down. Don't need to beat so fast. It'll divert blood away from skeletal muscle and toward the gut so that we can digest food and absorb nutrients. All right, so it's responsible for all of those day-to-day tasks that we need to do to keep the body going. Sympathetic division, fight or flight, it's responsible for basically amping us up and getting ready to respond to an immediate threat, an immediate stressor. Now, this weird modern world we live in causes a lot of us to be stressed all the time due to work or other situations. A lot of people currently are understandably very stressed about the pandemic, being home, maybe being out of work, things like that. Fear for loved ones, parents, things like that. So a lot of us are constantly in a state of stress, which leaves the sympathetic division constantly active. Maybe not fully active as if we had seen a zombie, you know, walking across our lawn late at night, but active to a greater degree than usual. All right, so the nervous system, we've already talked about this, but here's a simple chart showing that the central nervous system, uh, the nervous system is divided into the CNS or central nervous system and the peripheral nervous system or PNS. The central nervous system covers the brain and spinal cord. The peripheral nervous system is going to cover the spinal nerves and cranial nerves. Now, spinal nerves and cranial nerves, they can both be either motor or sensory. In fact, actually, most nerves, if I'm being technical here, are called mixed nerves, meaning they carry both motor and sensory fibers. But the individual fibers will be either motor or sensory. All right, so the sensory division, remember we had the somatic and the visceral divisions that aren't pictured in this schematic. The somatic senses come from, well, sensors that we're aware of, right? So temperature, pain, pressure, things like that in the skin, the muscles, the joints, bones, and then also the special senses such as sight, smell, taste, things like that. Those are all carried over cranial nerves, all right, to the central nervous system, to the brain. The peripheral nervous system also includes the motor division, which can be either somatic or autonomic, also known as visceral here. The somatic motor division, motor is going to be output. This is also known as the efferent division. And the somatic division of the motor division is going to control skeletal muscle. We can consciously do that. And then the motor division also has an autonomic branch, and it's going to affect the internal organs. Uh, it's going to affect cardiac muscle, smooth muscle, and glands. We're not consciously aware of it. We can't consciously control it. But there is a sympathetic division, which is fight or flight, and a parasympathetic division, which is rest and digest. All right. Whew. That is a lot of talking. Let me get some water real quick. All right. Yeah. So normally in class, we would watch this video. I will just recommend it to you. You can watch it on your own time. Crash Course Nervous System 1. The Crash Course Anatomy and Physiology videos, also a lot of the biology videos are relevant to our course. Um, they're great. They are fast, but within a couple of minutes, maybe 10, 15, the guy covers a lot of detail, and there's some great pictures to go along with it, some great animations to go along with it. So I'll just go ahead and recommend that to you. Um, if you have time, it may help you to watch these crash course videos. I'm not going to play his YouTube video in my YouTube video, right? It just doesn't make sense. All right, so let's talk about the neuroglia. All right, we talked about the neurons, plenty already. Neuroglia, these are the support cells, and there are 6 types. Ugh, sorry. They perform a variety of functions to support the neurons. They can bind them together. They provide a supportive framework for the nervous tissue, especially the astrocytes. We'll do that. You could also say that the ependymal and Schwann cells do that as well, providing a supportive framework. Wherever a mature neuron is not in synaptic contact with another cell, it's typically going to be covered in glial cells, right? Glial cells cover the neurons and thereby regulate their chemical environment. It also protects the neurons. It also means that neurons are only in contact with each other at places that are specialized for signal transmission, or in other words, where an axon, uh, synaptic knob meets a dendrite, right? So if you look at the picture down here, if we didn't have these Schwann cells, this is actually not a Schwann cell, this is an oligodendrocyte. If we didn't have these oligodendrocytes surrounding and insulating the axons here, okay, they could contact one another and they could actually transmit a signal from one neuron to the other at their axon. All right, and that actually wouldn't be good because neurons would just be, indiscriminately stimulating each other. For no good reason, they'd just be stimulating each other. And so the oligodendrocytes here produce the myelin sheath in the central nervous system, and they actually prevent that from happening so that only neurons that are supposed to stimulate each other or inhibit each other do so. All right. Neuroglia of the CNS. So you can see a couple of different ones here. In the central nervous system, there are four. We've got the oligodendrocyte here, which is going to produce the myelin sheath for the axons of neurons. Now, an oligodendrocyte is going to reach out to a couple of nearby axons. Whatever axons are nearby, it'll reach out and produce the myelin sheath. So here you see this oligodendrocyte is creating two segments of the myelin sheath for two different neurons. This one down here is reaching out to two different neurons, and it's actually creating three different segments. Here's one, here's two, here's three. So the oligodendrocytes will just reach out to whatever segments of the axon they can, whichever segments are nearby, and they'll produce that myelin sheath. All right, to surround and insulate the axon. We also have microglial cells, which are derived from macrophages. Well, actually, I think technically monocytes, but they act like a macrophage. They are immune cells that will engulf parasites. They will phagocytize parasites in the central nervous system. They also uh, just find waste and debris, broken down cell parts, things like that, and they'll break them down and recycle them. They clean up the tissue fluid, also known as cerebrospinal fluid in the central nervous system. All right, over here we have the astrocyte, which reaches out to nearby neurons and helps surround, cover, whatever the neurosoma, regulate its chemical environment, keep it from being indiscriminately stimulated. These segments that reach off of the astrocyte are called pedicels, or foot-like processes, feet for the astrocytes. And they reach out, they surround the neurosomas. They also, in some regions of the brain, will surround blood vessels, and the pedicels, or the foot processes of the astrocytes together, form part of the blood-brain barrier, which regulates what can get into the cerebrospinal fluid, what can get into the tissue fluid in the brain. And then lastly, we have the ependymal cells. All right, this is the fourth type in the central nervous system. The ependymal cells actually produce cerebrospinal fluid. They're the cells that actually produce it. Cerebrospinal fluid is basically a super filtered version of blood. I mean, really, all fluids in the body originally come from blood. Blood plasma, but this is a super filtered version of blood plasma. Some substances can readily cross the blood-brain barrier. Lipid-philic things like CO2, oxygen, O2, and alcohol actually can cross the blood-brain barrier, no problem. Just passes right through the lipids that make up these plasma membranes. Other substances can't readily get through, things like bicarbonate, that's key. Hydrogen ions also are key to regulating respiration. We'll talk about that in the respiratory unit. And sugar actually has to be pumped in. The brain runs on sugar. We mentioned the heart runs on mostly fat. It can metabolize virtually anything, but it prefers fat, the heart does. Cardiomyocytes do. The brain prefers sugar. In fact, it can't digest fat. The brain cannot run on fat. And so sugar has to be pumped in to the tissue fluid, has to be pumped across the blood-brain barrier to power the brain. Now, we also have this ketogenic diet, which is rising in popularity. It's a low carbohydrate diet. There's also the Atkins, which is low carbohydrate. And when you're on these diets, two things happen. If you're on a high-fat diet, like the ketogenic diet, you're going to produce ketone bodies, which the brain can actually metabolize for energy. doesn't want to. It wants sugar, but it can continue to function on ketone bodies. And then, of course, your liver can synthesize new glucose from other sources like, fatty acids and amino acids can actually turn those into new glucose to power the brain. All right, so on the Atkins diet and other low carbohydrate diets, the brain is not likely to die. You can convert other substances into sugar because the brain needs sugar. On the ketogenic diet specifically, you produce ketone bodies that the brain can run on. Doesn't want to, but it can. Um, yeah, so here we see a histological section of the brain. You can see the cell body here. You can see the nucleus at the center, and then all these tiny dark staining bodies. These are the nuclei of neuroglial cells that are surrounding the neurosoma here, and they're protecting it and insulating it. It's a blurry picture, a little hard to tell, but you can see there's plenty of them surrounding the neuron. So the types of neuroglia, we covered this in the picture before. If you want to, you can pause it right here and go ahead and read all this. But the four types found in the central nervous system are the oligodendrocytes that produce that myelin sheath, the ependymal cells, which produce the cerebrospinal fluid. They also have cilia on the apical surface that helps circulate. The cilia swirl and they help circulate the cerebrospinal fluid. Then you have the microglia, which are like small macrophages, which will eat pathogens, phagocytize them. And then finally the astrocytes, which they say are star-like. It's another one of those things where you have to squint really hard when you look at it and then believe with all your might. I don't think they look very much like stars at all, but they are named Astro for stars because they are supposed to look like stars. And they're the most abundant, as we said. They constitute over 90% of the tissue in some areas of the brain. And they form a supportive framework that fills in and creates the bulk in the brain. They also form a significant portion of the blood-brain barrier. And as we said, in the condition of multiple sclerosis, there are regions of the brain that have died, and astrocytes will actually migrate into that space and form scar tissue to fill in the dead region of the brain and really support the brain by filling in that empty region and give the brain some bulk. So the astrocyte, yes, it's another one of those things where you have to squint and believe with all your might. The oligodendrocytes kind of look like an octopus because they have all these processes that come off and they reach out to all the nearby axons, and they produce these segments of the myelin sheath in the central nervous system. You also have the microglia here, which are the white blood cells found in the central nervous system. And then finally the ependymal cells, which produce the cerebrospinal fluid. And then they have these apical cilia that swirl it around. In the peripheral nervous system, there are two more types, the Schwann cells and the satellite cells. Okay? Now, Schwann cells and satellite cells are only found in the peripheral nervous system. You will not find them in the central nervous system. Okay? So Schwann cells and satellite cells are only found in the spinal and cranial nerves. The astrocytes, oligodendrocytes, microglia, and ependymal cells, which are found in the central nervous system, will only be found in the central nervous system. You will not find them in the peripheral nervous system. All right, and so that division should help a little bit with remembering the 6 types of neuroglia. Also, the 2 that you find in the peripheral nervous system, this was just a little memory trick I used when I was a student. They both start with the letter S for whatever reason. I can remember if I can remember the names of all six, I said, well, there are two that start with S, and I can just remember those two are found in the peripheral nervous system. The other four are found in the central nervous system. Now, Schwann cells are also known as neurolimocytes. They produce the myelin sheath in the peripheral nervous system. You remember what cells produce the myelin sheath in the central nervous system? Right, that was the oligodendrocytes. The Schwann cells are doing that job in the peripheral nervous system. And they also, it says, assist in the regeneration of damaged fibers. We have a slide where we can show that happening. Um, the myelin sheath produced by the Schwann cells helps guide the neuron um, and to its target and repair the connection. We'll see the picture in a little bit. You also have the satellite cells, which surround the neurosomas in the ganglia of the peripheral nervous system, basically surround them, protect them, provide electrical insulation, and then also regulate their chemical environment. Kind of similar to the way astrocytes did in the central nervous system. All right, so mature neurons do not divide. They, if one is injured, the neuron will usually die. Um, in the peripheral nervous system, if a peripheral axon is injured, it can regenerate. The axon is separated from the cell body. Its myelin sheath will degenerate. Schwann cells, however, and the neurolemma will remain. So the remaining Schwann cells provide a guiding sheath for a growing axon or regrowing axon. And if the growing axon does actually establish the connection with its target cell, the function will return. But if not, the function will be lost. We will not be able to stimulate that target cell. That's in the peripheral nervous system. In the central nervous system, these axons lack a neurolemma, and so there's nothing there to they lack neurolemma. That is, they lack Schwann cells. There's nothing there to act as a guiding sheath, so the oligodendrocytes do not proliferate after injury, and then regeneration is unlikely. So what we're looking at here is in the peripheral nervous system. This is not likely to happen in the central nervous system. Here we have an axon for a neuron. There's been some injury, and the axon has been severed. Okay, now the axon is actually going to degenerate. It's going to degenerate and break down, and when that happens, this neuron, this is we're going to say a motor neuron, is not able to stimulate the skeletal muscle fiber or fibers that it used to stimulate. And so we actually won't be able to contract those muscle fibers. All right. However, the neurolemma and the Schwann cells, they remain. They form a tube here, okay, which is going to guide the regrowing axon back to the target, back to the site of the neuromuscular junction. Okay, so if the axon is able to follow this tube all the way back to the target cell where it originally formed a synapse with the target cell, then we can reestablish this connection, and this neuron will again be able to stimulate the skeletal muscle fiber or fibers that it used to stimulate. However, if the neurolemma, if this tube made of the Schwann cells degenerates, or if the axon is just simply not able to follow the tube to the target, then the neuron will probably die and we will never regain the ability to control this skeletal muscle fiber, and it will atrophy. All right, so here you can see the Schwann cells in the central nervous system. This is a unipolar neuron, by the way. We have one process coming off, so this is a sensory neuron in the peripheral nervous system. You've got the Schwann cells here, okay, producing that myelin sheath. Then you had the satellite cells, which surround the neurosoma, especially of the sensory neurons, whose cell body is located in the ganglia outside the spinal cord. All right, the two cell types, the two ganglia types in the peripheral nervous system, Schwann cell, satellite cell. Schwann cells produce the myelin sheath, very important for that reason. I feel like there was, okay, one more thing I wanted to say. Each segment, by the way, of the myelin sheath in the peripheral nervous system is one Schwann cell. So this segment is a Schwann cell that has wrapped itself again and again and again in layers around this region of the axon. Next segment down, that's a different Schwann cell that has wrapped itself around again and again and again around the axon. If we go back to the central nervous system, you can see the oligodendrocyte is a separate cell here which has reached out and formed several segments of the myelin sheath. Okay, and so an oligodendrocyte in the central nervous system also produces a myelin sheath, but it does so by reaching out to the axon and forming these segments. In the peripheral nervous system, each segment is a Schwann cell. All right, so the myelin sheath, it is an insulating layer around the nerve fiber. It's formed by the oligodendrocytes, as we said, in the CNS, Schwann cells in the PNS. And its composition is like other plasma membranes in general, about 20% protein, about 80% lipid. Myelination is the process of producing the myelin sheath, and it is not completed until late adolescence. That's one of the reasons that teenagers tend to be so impulsive. It's generally believed is because myelination is not complete in especially in the central nervous system. All right, so good. In the image here, you can see that the segment here is wrapping around the axon, and it's wrapping around inwardly. So as it grows, it pushes in. All right, so this is how myelination occurs with the oligodendrocytes in the central nervous system. In the peripheral nervous system, actually, when it's growing and it's producing new layers to wrap around the axon, it grows outwardly. All right, so we talked about the nodes of Ranvier before. These are the gaps between the segments of the myelin sheath. The gaps are called the nodes of Ranvier. The regions between the gaps are called the internodes, or between the nodes, internode, between the nodes. And you can see that here. You've got the nodes of Ranvier. Here you've got the internodes between them. Same over here, nodes of Ranvier, internode between. These are actually both oligodendrocytes. So two factors affect the rate of conductance along an axon, and that's going to be the diameter of the fiber and the presence or absence of a myelin sheath. And so the diameter of the fiber is very important. The greater the diameter of the fiber, the bigger it is across, the faster the signal is actually going to travel down the axon. And so you may have heard, if you read neurology journals, of the giant squid axon. And this is not the axon of the legendary giant squid. This is a giant axon of a regular squid. And the diameter of these fibers is quite large. Many of them are visible to the naked eye. And they have to run the entire length of the squid. They're very wide, they have a large diameter. And the reason for that is that when the squid senses danger, it needs to escape. And it does that by contracting its entire body and then propelling itself away. And in order to pull this off quickly and escape the predator, it needs to send this information very quickly from one end of its body to the other, and it's able to do that because it has axons with a large diameter. Neurologists often, well, scientists who study the nervous system often use uh, squid axons in their experiments because they have such a large diameter. Makes them interesting. Good. The presence or absence of myelin. Not all nerve fibers are going to be myelinated. Um, so in the peripheral nervous system, yes, they will be myelinated. In the central nervous system, there are some exceptions where they will not be myelinated. All right. So the axon hillock is that region where we initiate or trigger an action potential, and it contains a lot of voltage-gated sodium channels. The voltage-gated sodium channels, they remain closed when we're at resting potential, which is around -70 for a neuron. Um, However, they open when we reach threshold. All right. As the sodium enters the cell, we're going to depolarize it. And we talked about all this in the muscle unit already. But we're going to depolarize the cell as we, after reaching threshold, as we throw open the voltage-gated channels. Positively charged sodium floods into the cell, which makes it more positive, and we will continue depolarizing or flipping our polarity until we reach about positive 30 millivolts. All right. And then once we reach this point, once the membrane has become more positive, we are going to open potassium channels. And potassium channels are going to, voltage-gated potassium channels, are going to let potassium out of the cell, which makes it more negative. If we let positive charges out of the cell, that's going to make the cell more negative. And as that happens, the voltage is going to drop, or in other words, the charge across the membrane is going to drop, and the cell will repolarize. It will become polar again. As the membrane potential drops below -70 millivolts, it is temporarily hyperpolarized. In other words, we overshoot just a little bit when we are repolarizing. After that, the potassium channels will close, and we use the sodium-potassium pump, also known as the sodium-potassium ATPase, which is going to pump 3 sodiums out of the cell for every 2 potassiums it pumps in, and by pumping more positive charges out of the cell for each turn through the pump, we're going to be pumping more positively charged ions out, which makes the cell ultimately more negative over time. Well, it maintains the negativity of the cell. I'm sorry, it is getting very late. I'm going to try and make it through this before we call it quits for the night. But all right, so this maintains the concentration gradient. We also have leak channels, just as we did in the muscle, which is going to maintain the resting potential. All right, so we talked about this in the muscle unit, but we didn't maybe go into it in as much detail in regards to the axon. And so here we're looking at a segment of the axon. All right. We've begun an action potential here on the left, and we've thrown open voltage-gated sodium channels, which causes positive charges to flood into the cell, makes it more positive. That causes voltage-gated sodium channels in the next region to open up, and we're going to see sodium flood in, makes this region more positive. Well, guess what? That opens up voltage-gated channels in the next region over. Sodium floods in, and that makes this region more positive, all right, which is going to trigger voltage-gated sodium channels in the next region. And so this signal propagates down the length of the axon. It is conducted down the length of the axon, this action potential is, until we reach the synaptic knob, And there we will trigger voltage-gated calcium channels, which calcium comes in and then it binds to the vesicles and causes them to fuse and dump out their neurotransmitter. Now, an action potential is an all or none response. Basically, either you reach threshold and begin an action potential, or you don't reach threshold and you don't have any action potential. It is not graded. Graded means you can have varying levels of response. And with an action potential, either you have one or you don't. And you're not going to have big action potentials and little action potentials. You're going to have one action potential. It looks the same every time. They use the phrase, it is stereotyped, meaning it looks the same every time. All right, so if a neuron axon responds at all, it responds completely with the action potential. The nerve impulse is conducted whenever the stimulus reaches threshold intensity or above, and all impulses carried on an axon are the same strength. You cannot have super strong action potentials and weak action potentials. They're all the same. And then a stimulus of greater intensity may produce higher frequency of action potentials, but not stronger impulses. Basically, if you have a high intensity stimulus, you may get more action potentials. You may get, 1, 2, 3, 4, 5 action potentials, whereas a lower intensity stimulus that still reaches threshold might only get you like one action potential. You can increase the rate at which you have action potentials with a strong stimulus, but you will not get stronger action potentials. You can make them happen more often with a big stimulus, but you can't get bigger ones. They look the same every time. An action potential looks the same every time. All right, so during an impulse, the portion of the axon that is actively conducting the action potential is not able to respond to another threshold stimulus of the normal strength. And this is called the refractory period, which has two parts: the absolute refractory period and the relative refractory period. So during the absolute refractory period, this is the time when the threshold stimulus cannot generate another action potential. No matter how big the stimulus is, you cannot generate another action potential during the absolute refractory period. And the reason for that is that the voltage gated sodium channels are blocked. They are not able to respond. I don't know how deep I want to get into that. I'll just say the sodium channels cannot respond. The relative refractory period is the time when only high intensity stimuli can generate another action potential. So the repolarization is not yet complete, and the membrane is reestablishing its resting potential. The refractory period is going to limit the number of action potentials that are generated per second. So at the dendrites, we are going to see EPSPs and IPSPs. These are our excitatory postsynaptic potentials and inhibitory postsynaptic potentials. And as you might get from the name, excitatory postsynaptic potentials are excitatory potentials that occur after the synapse, or rather on the target cell. And in this case, we're talking about on the dendrites of a target neuron, the second neuron in a chain. For IPSPs, this signal will be inhibitory. It will inhibit the formation of an action potential. These are graded responses. We said action potentials are not graded. They are all or none. These are not action potentials. These are postsynaptic potentials. These are graded potentials, which means they can be big or small. All right, you can have a big excitatory postsynaptic potential. You can have a small excitatory postsynaptic potential. Same with the inhibitory postsynaptic potentials. And these are occurring again on the membrane of the dendrites of the target neuron. Okay. All right, so for excitatory postsynaptic potentials, basically we're going to release a neurotransmitter that binds to sodium channels. And when those sodium channels open, these are ligand-gated sodium channels, by the way. They're ligand-gated in that they only open when they bind the neurotransmitter that they specialize in. And the neurotransmitter binds to the sodium channels, opens them up. Some sodium floods in. All right. Now, the amount of sodium that floods in is going to depend on the number of channels we have open. And the number of channels we have open depends on the amount of neurotransmitter that is released. Okay? And so if we release a little bit of neurotransmitter, that's going to bind to a couple sodium channels. It'll make the cell more positive. But is it enough to reach threshold? Well, if we're only binding a couple ion channels, probably not. And then over time, the sodium-potassium pump is going to pump the sodium back out, and we go back to our resting membrane potential. If, however, we bind enough sodium channels, enough ligand-gated sodium channels, to have a big enough influx of sodium into the cell and make it positive enough, all right, that we reach threshold, then we can open voltage-gated sodium channels at the axon hillock and initiate an action potential down the axon. All right. So these excitatory postsynaptic potentials, they make action potentials more likely. On the other hand, you have inhibitory postsynaptic potentials, and these, when they bind, are going to open potassium channels or chloride channels. If you open a potassium channel and let potassium out of the cell, that's going to make it more negative. There's a lot of, also by the way, chloride outside of the cell. Sodium and chloride are at high concentration outside the cell. If you open chloride channels, negatively charged chloride will flood into the cell, making the cell more negative. And if we make the cell more negative, we take it further away from threshold and hyperpolarize the cell, and we make postsynaptic potentials. Aptic action potentials in the postsynaptic neuron less likely. So these EPSPs and IPSPs are added together in a process called summation, and you basically add up all of the excitatory stimuli and all of the inhibitory stimuli, and if at the end of that you can reach threshold, then you will depolarize and initiate an action potential. All right, so it's the summation of all inputs, and this usually occurs at the trigger zone of the axon hillock. All right. And so you can see here the resting membrane potential is about negative 70 millivolts. If we open potassium channels, that's going to make the cell more negative. Okay? If we open chloride channels, chloride's going to enter the cell, and that'll make it more negative. Take it down to minus 80, minus 90. All right? And that's what happens with inhibitory postsynaptic potentials. We make the cell more negative, and we take it further away from threshold, which is up here about 55, about negative 55 millivolts. All right? We increase the distance between our membrane charge and threshold, which means we're going to need a bigger excitatory stimulus to overcome that inhibitory stimulus. Okay? Now, an excitatory stimulus, on the other hand, brings you closer to threshold, makes the cell less negative or more positive by letting sodium in. And so we're going to get closer to threshold with an excitatory postsynaptic potential. And so you just have to add those up, the inhibitory postsynaptic potentials and the excitatory postsynaptic potentials, and see where the charge of the cell membrane is after all of those graded potentials. If we sum them up and we reach the threshold, negative 55 millivolts, then we will trigger an action potential by opening voltage-gated sodium channels in the axon hillock, and then we will become more positive until we reach about positive 30 millivolts. In this region, at C, or actually D, okay, D's over here. D should be on the left side toward the peak, just before the peak. We should open up potassium channels, which are going to make the cell more negative as we let positive charges out of the cell. All right, remember we talked about this in the muscle unit. We're just recapping here, but basically we overshoot a little bit, right? We make the cell more negative than the resting membrane potential, and that's called hyperpolarization. Depolarization is where we flip the polarity, make the cell more positive. Repolarization is where we go back to our resting membrane potential, except hyperpolarization is where we overshoot it a little bit. We go a little extra negative. And then just over time, the sodium potassium ATPase and the sodium and potassium leak channels will bring us back to our resting membrane potential, where we stay until a stimulus arrives, either excitatory or inhibitory. And we showed a little cartoon earlier that we drew. Just to come back to this again, if you see that the light has turned green, this first neuron is going to release excitatory neurotransmitters that stimulate the dendrites, that excite the dendrites on the second neuron. All right. On the other hand, you've got this pedestrian neuron, and if it sees pedestrians, it's going to release inhibitory neurotransmitters that are going to make the plasma membrane of the dendrites more negative and make it more difficult for this neuron, the gas pedal neuron, to fire. All right. Sorry, I need some water again real quick. All right, so that was almost all the hard stuff for today. We're going to finish up with some comparatively easy stuff. So on to diseases of the central nervous system. We've mentioned it a couple times here, multiple sclerosis. Sclerae, the word sclerae, refers to scars or lesions. And in the picture at the top right, you can see this dark red region. That is a sclera, singular, located within the brain, right? The cerebrum of the brain. And the cause is unknown, but it's believed that genetics contribute to the condition. It's diagnosed either by MRI or analyzing the cerebrospinal fluid. At the bottom right, we have an MRI where we are taking slices or sections of the brain. Now this is with imaging, we're not actually cutting the brain, but you can see these hot spots appearing. Those are the sclerae, the scarring that we're referring to. All right. So the signs and symptoms of multiple sclerosis are actually dependent on the location of the lesions. And so you know that different regions of the brain are specialized for different functions. If you have these sclerae pop up in the region responsible for speech, you will be unable to speak. If you have sclerae popping up in regions responsible for maintaining balance or body posture, you won't be able to maintain your balance or body posture anymore, or you will do so with greater difficulty. So because the signs and symptoms depend on the location of the sclerae, almost any neurological ailment could be due to multiple sclerosis, MS. All right, so here we have Parkinson's disease. Famous Louisvillian, Muhammad Ali on the left, suffered from Parkinson's disease as a result of the many brain injuries, concussions he suffered while boxing. On the right, Michael J. Fox, famous actor, also had Parkinson's disease. The two have worked together to raise funds for Parkinson's research. Muhammad Ali, early in his career, was really light on his feet, fast, powerful. Famously said, "Float like a butterfly, sting like a bee." That was him. And then he was a conscientious objector who did not go to war when he was drafted, and as a result, was not able to continue boxing. He was sort of kicked out for a number of years, and when he came back, he was older and not as fast. And what he figured out was that even though he wasn't as fast as some of the other boxers, wasn't as young as some of the other boxers, what he figured out was he was tougher than them. And so he'd let them just wail on his head for a couple of rounds, and then he'd step in and knock him out. All that trauma to the brain, though, resulted in the death of the dopaminergic cells, especially in the substantia nigra of the brain. Dopaminergic cells are responsible for a variety of functions, one of which is coordinating body movements. And so by dopaminergic cells, we're referring to the cells that release dopamine. They're also responsible for reward pathways and things like that in the brain. But here, in the case of Parkinson's disease, these cells are responsible for controlling body movements. There's a GIF or GIF down here showing a gentleman eating with a spoon. You can see with a regular spoon, he's suffering from Parkinson's, and the food on the end of the spoon is going everywhere due to the shake in his arm. This is a special medical spoon that has a hinge in the middle of it, which means when he shakes, the distal part of the spoon stays more or less in place in space. But you can see in motion sort of the characteristic shake of Parkinson's. People with Parkinson's, they have also difficulty seeing, as well as autonomic dysfunction, trouble sleeping, and mood-related issues. As we said, dopamine is really important to regulating mood as well. So the cause, just to go back to the beginning here, the cause is unknown, but it's suspected that genetics play a role. In the case of Michael J. Fox, who was not a boxer, that's likely the case. In the case of Muhammad Ali, it was repeated brain injury that resulted in the death of these cells. One just medically interesting thing, by the way, because this affects dopaminergic cells, there's another condition, well, that you've heard of called schizophrenia. Just every once in a while, a word will just not, it'll refuse to come to the forefront of your mind. It's on the tip of your tongue. Schizophrenia results from hyperactivity in the dopaminergic cells. And some medications used to treat schizophrenia downregulate the activity of the dopaminergic cells to the point that it causes Parkinson's-like symptoms. Just a strange medical thing there. So that was diseases of the central nervous system. We've got a couple diseases of the peripheral nervous system I'd like to cover. We've got nerve damage. This is due to trauma. And then Guillain-Barré, and then Zika and dengue. Remember Zika? Zika was awful. I mean, Corona's awful. Corona results in death. Zika resulted in malformation of the brain and affected babies, innocent babies. Sciatica, man. Yeah, I've got sciatica. It is a pain, let me tell you. So here we see the path of the sciatic nerve traveling through the greater sciatic notch of the pelvis here, and then down the femur. All right. Now, actually, the nerve roots that give rise to the sciatic nerve are going to be located up here around the level of L3, L4, or no, L4, L5, but close up here. The actual nerve roots that give rise to the sciatic nerve are going to be up here. And frequently what happens is that you have a bulging disc which presses on the nerve roots and compresses them. and that results in the sensation of numbness or pain that travels down the path of the sciatic nerve, down the back of the leg, and then the sciatic nerve above the knee splits into the tibial and fibular nerves, and so the pain will actually travel down through the sural region and then finally into the foot. Uh, can result in severe back pain. And just to give you my personal experience, I've been taking muscle relaxers for a little while now, doing stretches, changing the way I sleep and sit, and all this to try and take pressure off of the spine and the bulging disc and all this. Went to see a chiropractor, and I'm getting better. And at this point, I can stand and walk for about 10 to 15 minutes without uh, getting into some severe pain. Before seeking treatment, it was so bad I couldn't stand, sit, walk, anything. It was constant, excruciating pain. All right, and so I'm hoping, because it's getting better over time, hoping it'll just continue uh, down that path. Next up is the funny bone. You've heard of this. You hit your elbow, you say you hit your funny bone. What did you hit? Well, it actually wasn't the funny bone. It was the ulnar nerve. All right, so the ulnar nerve travels down the brachial region, all right, sort of next to or behind the humerus, and then it passes behind the medial epicondyle. Remember, the medial epicondyle of the humerus was this flared out bit of bone, which is going to serve as a point of attachment for muscles. And um, the ulnar nerve, as it passes behind the medial epicondyle, is vulnerable. Basically, it's up against bone here, and if you were to hit your elbow here, you're going to compress the ulnar nerve. And this leads to, a sensation of numbness or pain that's usually temporary. My wife has a propensity for hitting the funny bone, and she was working an office job a number of years ago and hit it on some sharp edge on a desk or on a filing cabinet or something. And she actually experienced pain from it for about 6 months and had to go see a neurophysiologist um, to get it assessed. Did eventually get better on its own, but that numbness and pain lasted 6 months. It was unpleasant. Next up, we have denervation atrophy. We mentioned this briefly at the end of the muscle tissue unit. And so whenever a nerve is severed, paralysis, it says may occur, but actually will occur because skeletal muscle never contracts unless it's stimulated by a nerve. All right. So if you sever the nerve, then you are unable to stimulate the muscle and therefore unable to contract it. And as you are unable to contract it, your body will replace the muscle fibers with fibers made of connective tissue. And when that occurs, you can't replace the connective tissue later with muscle fibers. And in the case of denervation atrophy, you've severed the you've severed the nerve connection to the muscle. And so there's really nothing you can do to reverse that, and there's nothing that you can do to prevent it except not sever the nerve in the first place. In this individual, you can see the flattened calf on his right sural region, and that is because over time the gastrocnemius and the soleus have atrophied and been replaced with fibrous connective tissue. All right, now Guillain-Barré syndrome is an interesting syndrome, and it actually can be caused by infection with the Zika virus or the dengue virus. The two are related. And Guillain-Barré can actually follow infection with a number of different viruses. It's almost always a virus, and it's frequently Zika or dengue, and the two are related, but it can occur with other infections as well. All right. And so we say here the viral infection may even clear before the onset of Guillain-Barré. What is Guillain-Barré? It is an autoimmune condition that results from an infection. Basically, you have an infection, and it could even be a mild case of dengue fever. Could be a mild case of dengue fever. In Zika, if you remember, in adults, typically had a very mild fever, not a very serious disease in adults. However, it caused, developmental deformities and developmental issues in a fetus, and especially in the cranium of the fetus, the brain and skull of the fetus. But in adults who were not pregnant, typically very mild symptoms. All right, and so you can have a very mild infection with Zika virus, but if your immune system becomes hypersensitized to its presence after you've cleared it, the immune system may go on attacking. And in the case of Guillain-Barré syndrome, the immune system has continued on attacking the nerves of the peripheral nervous system. Now, onset of Guillain-Barré can take a few hours or days. The symptoms can remain for weeks or months, and it typically presents as an ascending weakness that may result in respiratory failure. Now, we say here it may remain for weeks or months. Just to be clear, Guillain-Barré is sometimes fatal, and it can last longer than weeks or months as well. If the damage to the central nervous system, if the damage to the peripheral nervous system is severe, that can result in not only muscle weakness, but paralysis that lasts years. I have seen on TV the case of a few people with Guillain-Barré who have, they spent the next year in a hospital bed, and once they started getting better, still they had to go through physical therapy to relearn how to control the muscles. All right, so drugs that affect the central nervous system, we're going to talk about cocaine, methamphetamine, cannabinoids, and opiates. So, comparing neurotransmitters and hormones, neurotransmitters are released by a neuron and they act very quickly. Hormones, on the other hand, are released by endocrine glands and they tend to take longer to act because they have to travel through the bloodstream. But some chemicals can be used as both a neurotransmitter and a hormone. So the definition for either really is just flatly a neurotransmitter is released by a neuron and a hormone is released by a gland. So you can take, for instance, norepinephrine. Norepinephrine is released by neurons as a neurotransmitter, and then it can also be released by the adrenal gland as a hormone. All right. So we're not just, by the way, talking about drugs, we're also talking about neurotransmitters. So serotonin is a neurotransmitter, and it's involved in many pathways, especially those involved in happiness and anxiety. A lack of serotonin is the most common cause of major depression. And then we convert serotonin to melatonin, which prepares the brain for sleep. A lot of people these days are taking over-the-counter melatonin, which helps you sleep. Many people find that it helps them sleep. You produce melatonin naturally. Your brain, specifically the pineal gland of the brain, will convert serotonin to melatonin. All right. There is a condition called seasonal affective disorder where the pineal gland is overactive and converts too much serotonin into melatonin. I believe we'll talk about that in a later unit. But anyway, when you are depressed, your doctor will probably prescribe to you an SSRI, which is a selective serotonin reuptake inhibitor, which keeps serotonin in the synapse for longer to keep stimulating the postsynaptic neuron as if you had more serotonin than you actually have. All right, and that'll elevate the mood. All right, now norepinephrine is another neurotransmitter and it creates a sense of well-being in the brain. Dopamine, as we mentioned, is responsible for reward pathways and pleasure, happiness. A lack of norepinephrine and dopamine can create, a lack of dopamine can create, depression. A lack of norepinephrine can create anxiety. Then we have GABA, also known as gamma aminobutyric acid, which is a modified amino acid. And it has inhibitory actions. When it's released, it will inhibit neurons, and it actually acts to calm down neurons. So releasing GABA calms down overactive neurons. We also have opioids, um, also known as endorphins, which are going to suppress pain. So we first discovered opium, which is produced by the poppy plant. We extract opium from the poppy plant. And we derived from opium a drug called morphine. And by the way, whenever you discover a natural substance that actually works in the body or the brain, um, it does so whenever it has an effect in the brain, it does so by activating receptors that were already present in the brain, right? So we developed morphine, and the question becomes, how does morphine work? Well, it turns out there are receptors for morphine in the brain. Now, why would there be receptors for morphine in the brain? Well, we produce a neurotransmitter, or a class of neurotransmitters, that we call endorphins. All right, now endorphin stands for endogenous morphine. All right, endogenous means naturally made within the body. So endogenous morphine, these are substances similar to morphine that are produced within the body in small amounts, um, produced in the brain in small amounts, and they bind to the receptors, mu opioid receptors, and they suppress pain. That's why morphine works. It binds to the same naturally occurring receptors in the brain and shuts down pain. Whenever you are experiencing pain due to strenuous exercise or injury, you will release endorphins in the brain to help suppress it, protect yourself from the pain. It's obviously not foolproof. We do still experience plenty of pain in our lives. Then lastly, we have the endocannabinoids, and as you might have guessed from this name, these are cannabinoid substances that are produced endogenously in the brain. And that's why cannabinoids, THC, and possibly uh, CBD have an effect in the brain. The research really isn't done on CBD yet. There's still plenty of research to go, and a lot of the supplements they're selling you on the street or at the gas station or wherever else it's just crap. But THC has an effect in the brain because there are endocannabinoid receptors in the brain. Our brain naturally produces cannabinoid substances that will stimulate the endocannabinoid receptors, and they control, of course, you guessed it, appetite, mood, and memory. All right, so we mentioned earlier the most common type of drug used to treat depression is an SSRI, or selective serotonin reuptake inhibitor. What is reuptake? Well, that is when we have released a neurotransmitter into the synaptic cleft, and now we need to stop stimulating the second neuron. And so we are going to take it back up into the first neuron's synaptic knob. All right. Now, this drug is called a selective serotonin reuptake inhibitor because it selectively inhibits reuptake at serotonin neurons. It is selective for serotonin synapses, and it is going to block reuptake. All right. So if you block reuptake, you take the presynaptic neuron and you prevent it from taking back up the serotonin. That causes the serotonin to be present in the synapse for longer, and that allows us to continue stimulating the postsynaptic neuron or the second neuron for longer. Kind of makes the brain act like it has more serotonin than it actually does. So an example is Prozac, or generic name fluoxetine. This is an SSRI. SSRIs are typically better than MAOIs. You may have heard of them. They are monoamine oxidase inhibitors. Now, they inhibit the enzyme monoamine oxidase that breaks down serotonin in the synapse. You remember acetylcholinesterase from acetylcholine synapses? Acetylcholinesterase broke down acetylcholine to remove it from the synapse or synaptic cleft. At serotonin synapses, we have another enzyme called monoamine oxidase, which is going to break down serotonin and remove it from the synapse. A monoamine oxidase inhibitor is going to block that enzyme, monoamine oxidase, and prevent it from breaking down the serotonin, which keeps serotonin in the synapse for longer, which helps us stimulate the postsynaptic neuron longer and elevate our mood. But it's used maybe not as a last line of defense, but it's definitely not your first choice for depression because the side effects are more widespread. Monoamine oxidase is used to break down not just serotonin at serotonin synapses, it also breaks down norepinephrine at norepinephrine synapses and dopamine at dopamine synapses. And so if you inhibit this enzyme, monoamine oxidase, at all of those different types of synapses, you're going to see a lot of different side effects. Selective serotonin reuptake inhibitors are selective for serotonin synapses, and so they're more targeted. They're generally your first choice. Now here we're talking about cocaine and methamphetamine. Both drugs are going to work on neurons that utilize norepinephrine, but also dopamine and serotonin. And so methamphetamine is going to displace norepinephrine from the synaptic vesicles. You see these synaptic vesicles down here? All right, so we are going to, I'm sorry, it's tired. Let me get my thoughts together. Methamphetamine is going to displace norepinephrine from the synaptic vesicle, basically force it out of the synaptic vesicle and force it to leak into the synaptic cleft. Okay. Cocaine is going to block reuptake. Now, methamphetamine also blocks reuptake, right? So reuptake is where we take it out of the synapse, bring it back up into the presynaptic neuron. Cocaine blocks reuptake of serotonin, dopamine, and norepinephrine, so we keep stimulating these postsynaptic neurons for longer. All right, creates that sense of well-being associated with cocaine. Overstimulating the norepinephrine neurons, though, can also create paranoia, which is common with methamphetamine use. Methamphetamine use not only prevents reuptake, it also forces leak of the norepinephrine into the synapse. But in both cases, norepinephrine spends a greater amount of time, as well as dopamine and serotonin, in the synapse, so these secondary or postsynaptic neurons get stimulated for longer. Now we say it occurs at dopaminergic and serotonin synapses as well. Primarily acts at norepinephrine synapses. All right, so drugs that affect the peripheral nervous system. We're going to talk about acetylcholine, acetylcholinesterase, nicotine, muscarine, atropine, and neostigmine. Acetylcholine, we've talked about. It's used to control the skeletal muscle. It's also used in some synapses of the autonomic nervous system. And I say some, but it really should be many synapses of the autonomic nervous system. And acetylcholine has to be broken down in the synapse by an enzyme called acetylcholinesterase, which is abbreviated ACHE or ACE. All right, now there are two types of acetylcholine receptors known as nicotinic and muscarinic receptors. Nicotinic receptors are located at the neuromuscular junctions. That is the junction between the neuron and the muscle fiber, the skeletal muscle fiber. We also find them, nicotinic receptors, at some of the synapses in the autonomic system. All right. Now, these synapses can also be activated by nicotine, right? So they're called nicotinic because nicotine can bind to the nicotinic receptors for acetylcholine. Muscarinic receptors are found at some synapses of the autonomic nervous system, and they can also be activated by muscarine, which is found in a certain kind of fungi. So let's open Paint back up. I always draw this as... Alright, and then let's find another shape. Alright, and then where's the fill here? Yeah. Why is that not working? Fill, solid color. I gotta use the paint bucket over here. Okay. So let's say this is acetylcholine. Alright, so it has two ends. It has a nicotinic end and a muscarinic end. All right, and so there are two different types of receptors. The nicotinic receptor is going to be shaped something like this. All right, and so the nicotinic end will fit in here. Let's go ahead and say nicotine is also kind of square shaped. Muscarine we'll say is round, and so the muscarinic end will fit into a curved receptor. All right, and so this is the plasma membrane here. This is a terrible picture. All right, so this is the plasma membrane. These receptors are embedded in the plasma membrane. The nicotinic end of acetylcholine, also nicotine, will bind the nicotinic receptors. The muscarinic end of acetylcholine, also the molecule muscarine, will bind the muscarinic receptors. Now, the difference between the two is that if you remember from the muscle unit, the nicotinic receptor located at the neuromuscular junction, also certain parts of the autonomic nervous system, is a sodium channel, or actually it's an ion channel. So sodium can travel through, mostly sodium. Also, a little bit of potassium can travel through as well. Okay. The muscarinic receptor, on the other hand, is a G protein coupled receptor. And so basically when muscarine binds here, all right, to the receptor, that's going to activate a protein cascade on the other side of the membrane, which causes like a chain reaction of things to happen. So the two operate differently, and that's why we go to the trouble of talking about them separately if they both respond to acetylcholine. We can also target certain receptors by using drugs that simulate nicotine or muscarine also, or block those receptors also. So with nicotine poisoning, this usually results from the ingestion of tobacco or liquid nicotine. And in an adult, it would pretty much have to be pure liquid nicotine, like you're mixing up your own e-liquid, and not being careful and wearing gloves, that kind of thing. Your store-bought e-liquids are generally not going to have enough nicotine to make an adult sick, but they can make children sick. I think we mentioned before that tobacco plants evolved to produce nicotine actually as a pesticide to kill bugs that might try and eat them. So the first phase of nicotine poisoning is stimulatory, and so you're going to see muscle twitch, as well as nausea, tremor, hypertension, and tachycardia or a fast heart rate. The second phase of nicotine poisoning is a depressor phase, and so all of these target tissues are overstimulated and we can't stimulate them anymore. And so we're going to see paralysis followed by hypotension, that's low blood pressure, bradycardia, slow heart rate, and respiratory failure because the muscles that control respiration are skeletal muscles, which again respond to nicotine, or rather contain nicotinic acetylcholine receptors. So to treat them, you can treat with atropine for the bradycardia or the slow heart rate, and intravenous fluids for the hypotension. Atropine actually it kind of resembles um, muscarine, and so it can be used to stimulate the muscarinic receptors located on the pacemaker cells of the heart without stimulating further the nicotinic receptors located throughout the body. So that's why atropine is useful here. Myasthenia gravis, we mentioned before, is a chronic condition caused by an autoimmune disorder that targets nicotinic receptors at the neuromuscular junction. This leads to fewer receptors, which means it's more difficult to signal the muscles to contract. It's generally characterized by muscle weakness, but not exhaustion. People with myasthenia gravis are not tired, they're just not able to tell the muscle to contract. The first sign is typically weakness of the muscles that raise the upper eyelid, the levator palpebrae muscles, superioris muscle. As the condition progresses, though, weakness will be seen in other muscle groups, possibly leading to respiratory failure. In the end, it can be fatal. So you treat myasthenia gravis with neostigmine, which inhibits acetylcholinesterase. If you inhibit acetylcholinesterase, that means there's going to be more acetylcholine available in the synaptic cleft. More acetylcholine means you're going to get a more chance to stimulate the acetylcholine receptors and then initiate muscle contraction. All right. So neostigmine, though, is not specific for acetylcholinesterase at the nicotinic receptors. Neostigmine will act at all acetylcholine receptors. It'll inhibit acetylcholinesterase at all acetylcholine receptors. And so if you do that at the muscarinic receptors, you inhibit acetylcholinesterase, especially those found in the heart. You are going to speed up the heart. Well, no, that's going to cause, bradycardia, slow the heart down. So to prevent the bradycardia, the atropine must also be administered because atropine is going to stimulate the muscarinic receptors in the heart. Not stimulate, sorry, it blocks the receptors in the heart, which prevents the slowing down of the heart. It's kind of counterintuitive. Normally, muscarinic receptors in the heart are stimulated to slow down the heart. Atropine blocks those receptors and keeps them from getting stimulated. Sorry, it's kind of backwards and counterintuitive. The nicotine addiction example, so how do you get addicted to substances, especially nicotine? So when nicotine causes acetylcholine receptors in the brain to fire, the dopamine also gets released. And the dopamine is responsible for this feel-good feeling. It's also part of reward and behavior pathways. And so every time you do something that releases dopamine, you are then sort of training yourself to do it again and again to get that dopamine release. The nicotine binding also prevents the recycling of acetylcholine receptors, and so as time goes on, the older receptors they change shape. They become non-functional due to just sort of a normal wear and tear. So more nicotine is required to achieve the same result because there are fewer functional acetylcholine receptors. All right, so here's another crash course video. Feel free to watch that. All right, that's it. I just wanted to share this image. Keep calm and yo GABA GABA. GABA, of course, was gamma amino butyric acid, one of the neurotransmitters. All right, so that is it for today, and we've got 3 more PowerPoints to go through, and then we will be done for the semester. As always, feel free to send me an email with any questions you have. Thank you.